Okay, good morning. We are going to go ahead and get started on the meiosis lecture. And so we had already talked about mitosis in our last chapter. We're going to be looking at meiosis, which is different. Let's just review real quickly some facts about mitosis. So for one, in what kind of cells does this process occur? Well, for mitosis, that's all normal body cells. Why does that happen? Well, to make more cells for growth or repair. Which of the following separate during anaphase? You separate out your what? Your homologous chromosomes. And how many daughter cells are present at the end of the process? 2. And are the made cells haploid or diploid? They are diploid. And when you compare the daughter cells, are they identical or unique? They are identical, and crossing over and independent assortment does not occur. Oh, did I say homologous chromatids? Sister chromatids. Okay, so you separate your sister chromatids, that's right. Okay, so now we need to talk about meiosis. That is, when does meiosis going to happen? It's when you want to make reproductive cells. So we inherit traits from our parents, and those traits are controlled by genes on the chromosomes we inherit. But we are different than our parents, which arguably is a good thing. That is due to genetic variation that is created when these gametes, those reproductive cells, are produced during meiosis, and how those fuse, because that is a random joining of two different gametes. You have a lot of different possible combinations of outcomes, which means we're different from each other. Meiosis divides the DNA in a parent nucleus into 4 daughter nuclei that are not genetically identical to one another. Unlike mitosis, which made 2 genetically identical cells, this makes 4 genetically not identical cells. This is used in animals to make gametes for sexual reproduction, and male and female gametes fuse during fertilization to form a zygote. That is one single cell that's diploid, that has the full complement of genetic information that can develop into a new human. So in a diploid organism, i.e. us, those reproductive cells go through meiosis to make haploid gametes that are genetically unique. This cuts the number of chromosomes in half. So that means for humans, in a haploid gamete, we would only have 23 chromosomes. If you're talking about something like, I believe a horse maybe has 60 chromosomes, their gametes would have 30 chromosomes. So 30 plus 30 would then equal 60. So those 2 haploid gametes fuse and make that diploid zygote. You once again restore the normal number of chromosomes. Then that zygote divides by mitosis and makes a fully diploid organism. Okay, some overview. Remember, the goal is to go from 1 diploid parent cell to 4 different haploid daughter cells. Meiosis is divided into 2 steps. Now, you may remember from other classes, this is somewhat glossed over and really kind of described a lot like mitosis happening twice. And in some ways, that's true. There will be a few little more nuanced details here. So meiosis 1, or the first series of divisions, separates the homologs of each homologous chromosome pair, right? So we want to separate our homologs, our 2 chromosomes. Those are 2 chromosomes. There's our same 2 chromosomes. Here they're not duplicated into sister chromatids. Here they are. And those are going to be separated from each other. Now, you inherited this from one parent and that from a parent. Those are going to get separated during meiosis 1. And that then causes those cells to be haploid. The second series of things that are going to happen in meiosis 2 separate out the 2 sister chromatids. So now we have 4 different cells. Now, that's not the full story. We'll talk about some details in here. All right, so meiosis 1, to reiterate, separates the homologs, right? The 2 chromosomes. Remember, they pair your chromosomes of your 46 chromosome, you have 23 pairs. Each one of those pairs are homologs, one from one parent, one from the other parent. They both contain the same genes. Those are separated during meiosis 1. And then during meiosis 2, those sister chromatids are separated. Those copies that are linked together at the kinetochore that we talked about earlier with the mTOX separating them and all that other stuff. Okay, so first off, some things to watch. Watch the chromosomes, whether they're homologous, they're sister chromatids, and watch how they get separated. Once again, we have mTOX, or those spindle fibers that pull these apart. And let's go check this out. Okay, so in meiosis 1, all right, there can create a certain amount of genetic variation, which is what we want. All right, now remember, we're still starting off in here during interphase, during G2. We've already duplicated our sister chromatids and created those during the S phase, right? The cell is diploid because we have both homologous chromosomes for each pair, all right? And so we're now ready to begin. Going into prophase 1, we use the same stages, prophase, metaphase, anaphase, and telophase, to describe the stages that happen during meiosis. We just call it, prophase 1, or prophase 2. All right, so this is actually a really important part on prophase 1. Now I want to take a moment to talk about this. Homologous chromosomes pair up and crossing over happens. Now we'll talk a bit more about this crossing over once we're done with all the stages. Crossing over is literally where the actual homologous chromosomes cross over each other, and they actually break off pieces of each other and start swapping genes, big sections of genes. This induces a higher rate of genetic variation. So let's look at this in this image. Let's say the blue is from your mother and the red is from your father. Now, if we just separated those out, you would give 100% all, let's say you could create a gamete with 100% all of the exact same genetic information on this chromosome from your father to your descendant. That's not how it works. Because they do this crossing over, some of the genes carried by your mother and some of the genes carried by your father swap. And so those sister chromatids, once they separate out, are not going to be genetically identical, which induces more variation. Remember, variation is really good. We want variation. Variation is, wonderful in nature. And so we want to continue to have that variation. We'll talk more about this specifically in a little bit. But I want to just point out that's a pretty important stage that is very different from mitosis, because in mitosis, you want everyone to be exactly the same. That's the whole purpose of it. Okay, the purpose of this is different. So our chromosomes, just like the rest of prophase, like we saw with regular mitosis, your chromosomes condense. They wrap up from this loose chromatin. They form these tight chromosomes. Okay, we start to have our mTOX spindle fibers form. We get rid of the nuclear membrane, just like in mitosis. This prophase 1 crossing over is a big deal. You will need to pay attention to that. Okay, we've got metaphase, same things that happen with regular metaphase. You line up along the middle of the cell just like before, but here's where we differ a little bit. Okay, instead of just separating the sister chromatids, remember we separated the sister chromatids in mitosis, the two little pairs that were stuck together, and separate them. Instead, those homologous chromosome pairs. Remember, this is, let's say, chromosome 1 from your 1, and this is chromosome 1. Remember, they occur in pairs. Those are the same chromosome. Okay, this one I had gotten from my mother, this one I'd gotten from my father, but now they got a little blended during crossing over. But what we're going to do now is separate that homologous or homolog from this homologous chromosome, this homolog, right? We're going to separate those two homologs. Second metaphase, we're going to separate the sister chromatids, right? So this is a little bit more complex. So the arrangement of the homologs is random, and so that creates independent assortment. So these line up randomly, first pair, it's not first pair, second pair, third pair, fourth pair, they're all lined up. They might be oriented this way, they might be oriented this way, and then they're pulled apart. This creates some what's called independent assortment. This side doesn't get all of your mom's, predominantly mother's chromosomes, homologs. This side does not get all of your father's homologs. They are independently separated. Each one just gets lined up and they get handed off one way or the other. Because it's random, that creates a lot of genetic variation. Okay, anaphase 1, those homologous chromosomes are separated. Now look, we're pulling apart these homologous pairs, but there's this blending, these different colors that are occurring. That was because of crossing over. Okay, so this is what makes the first daughter nuclei haploid, is because they only have one version of each chromosome type, right? So this is for our first chromosome pair. This is our second chromosome pair. We should have 23 pairs. Those get separated from their pairings, and then that makes you haploid. But we still have sister chromatids. We still have these copies of each other, which are not exact copies at this point. Okay, so then we need to go ahead and telophase and cytokinesis. So once again, that actually breaks apart. There is some variation on how they move from one to another, depending on the organism. All right, so let's move on to this. All right, so far, okay, what type of process does mitosis, somatic cells, which are normal body cells, and meiosis is only reproductive or sex cells, gametes. Let's see, sister chromatids are separated in mitosis. Meiosis 1, you separate your homologous chromosomes. You have two cells. These are now haploid, not diploid. The daughter cells are unique. They are not the same anymore because of independent assortment and crossing over. And yes, both of those things occur. Meiosis 2 is a lot like mitosis. Okay, it separates these sister chromatids just like mitosis does. But you're starting with two already different haploid cells. Okay, each chromosome is still duplicated because of crossing over. They're not the same. Okay, so prophase, we get rid of any membrane. We start to form our spindle fibers. Prometaphase, we start to line up. Metaphase, we line up along the center. Okay, and I bet we're going to do an anaphase, the same thing that we do in mitosis, which is to pull those sister chromatids apart. Now, remember, I've got all these different options at this point. Okay, each chromatid now gets called its own individual chromosome. Right, during telophase 2 and cytokinesis, we split into two. Each of the original cells split into two daughter cells. Now we have 4 total haploid daughter cells. They are not genetically identical to one another, nor are they genetically identical to the parent. You decondense them and you're ready to go ahead and use them. Right? And so the big difference here that you're going to probably need to pay attention to is in meiosis 2, we separate our homologous chromosomes, meiosis 1, we separate our homologous chromosomes. In meiosis 2, we separate our sister chromatids. The number of daughter cells present differs, and then we talk about our haploid and unique, and no crossing over and independent assortment do not occur during meiosis 2. In fact, this chart, if you want to pick a chart to study, this would be a very, very good chart. As it highlights probably in the most condensed manner the differences between mitosis and the different stages of meiosis. Okay, so genetic variation, we're just going to cover this real briefly. Why is it important? Well, because we want to have variation. Multiple versions or alleles of the same gene present in a species. Here's a good example. We've got these homologous chromosomes. This was from one parent. This was from another parent. Okay, this is the gene that controls sickle cell disease. It's a protein called beta globin. If you have normal beta globin, right, then you have a healthy allele. This one would be the diseased allele, right? And so Having genetic variation allows us, that's just an example, having genetic variation allows us to have multiple varieties of alleles, and that as a species, then you would accumulate genetic diseases if due to inbreeding if you didn't have variation here. Everybody would have sickle cell, let's say. Cheetahs are a good example of inbreeding. There was very small population of cheetahs, and some populations have real problems with recessive genetic disorders because they don't have enough variation. So how do we induce genetic variation naturally? So one, crossing over. Here's crossing over. Here's a good diagram. So we actually lay across each other, and I'm going to give you this part here, and you're going to take this part there, and we're going to get some variety by mixing up parts of our alleles on our gene. Independent assortment. Okay, here we have your fathers. Here you have your mothers. And now we've swapped them around. We could end up with some differences in our gametes just to begin with due to independent assortment by how we separate those. Each pair is separated independently of one another. And then also during fertilization, you have a huge variety of possible outcomes for this gamete's genetic makeup. So by the time you look at uniting those two gametes, that also creates a lot of variation. All right, so first off, on crossing over, once again, the chromosomes in a homologous pair are close together. Okay, so you got these from your mother, these from your father, right? Then these sister chromatids are the copies of each particular chromosome. They can cross over, and I can pass you a little bit of my DNA. Let's cross over, and now this part is going to link to this chromosome. Now your chromatid here has mainly DNA from your mother mixed in with DNA from your father. This one, which has mainly paternal or father's DNA, now has a bunch of your mom's DNA on it. And we don't know exactly what gene, we could look, but we don't know exactly what genes are here. Okay, so that creates if you inherit this one as opposed if you inheriting this one, you will have a different genetic makeup depending on which one you inherit. All right, so they actually swap their DNA. This is really interesting. The DNA double helix is broken at the same place in non-sister chromatids. So that whole DNA polymer strand lays across one another, and they just exchange, right? Which is really, really interesting. Okay, so that's what crossing over would look like. You may want to take a moment to kind of try to track this down, looking at the letters and how it gets swapped around. Okay, independent assortment is just how we can separate our sister chromatids. Excuse me, I shouldn't yawn like on the camera or anything. One of the interesting things, I don't know if they say this, there's a bunch of different, let's just leave it this. We don't have necessarily numbers for this right now, but because of crossing over, there's a huge amount of potential genetic variation that will happen simply due to crossing over. On top of that, independent assortment by how do we separate each maternal and paternal homologous chromosome? There's about 8.4 million possible combinations for our 23 chromosomes. Just in this. Okay, this does not include any sort of crossing over. So that's a lot of genetic variation. Right? So if we would separate these due to independent assortment, we have this arrangement. Okay? There's no sister chromatids. Okay, now we have all of these different possible combinations, right? And so there's a lot of genetic variation. You can see they're not all the same. On top of this, we have fertilization. Okay, depending on which male gamete, which female gamete unite, that's all random. Okay, so due to crossing over, independent assortment, and random fertilization, the number of possible allele combinations in offspring is tremendous. Okay, so through independent assortment and fertilization, there's greater than 70 trillion different possible combinations of human zygotes. 70 trillion. So that's like a lot. That's like 70 million millions. And on top of that, that does not take into account the mixing that occurs due to crossing over. So that is why you and maybe your siblings that come from the same parent or big family, you've got quite a few kids from the same parents, they are not the same because these processes occur every single time. And even though they might have some similar characteristics, they definitely are not the same. Now, if you start talking about twins, if you're talking about identical twins, you take this one fertilized zygote and it splits, and then those two new cells develop into individual humans. So they are genetically identical. But on fraternal twins, you have two different eggs with two different sperm that unite and form two different unique zygotes. So they are not going to be exactly the same. Okay, there can be errors. Primarily, there's something called non-disjunction. It was when they don't sort correctly, and then you'll end up with odd number of chromosomes on your gametes. And you can have here, right? If a gamete with one extra copy fuses with a normal gamete, you could end up with trisomy 21. I believe that would be Down syndrome. Okay, here looks like we've lost. We have monosomy instead of trisomy, and we only have the X chromosome, so this would be Turner syndrome is what that's known as. So this is some different examples of how often that can happen, depending upon the frequency in a population. So you can see that trisomy 21 or Down syndrome is about 1 in 700. So that's not uncommon. Okay. All right, we will go ahead and stop there. And that was this whole chapter. So we moved very quickly. Take the time to study the chart that goes over everything and spend some time kind of looking at independent assortment and crossing over.